Billions of litres of cow's milk are produced every day around the world, but that number is about to change radically thanks to biotechnology and what is now called biotech cow's milk, produced artificially. But how can fresh milk, ready to drink, be made in a lab? To find out, we step inside the laboratories working to create this milk of the future. You might be wondering when this product first came to life. It all began with advances in cellular biotechnology. Back in the early 2000s, researchers were already cultivating human tissue for transplants and exploring how cells could produce complex compounds outside the body. The idea of using cells to make food was not entirely new. Cultivated meat, for example, was already on the radar. But milk, with its unique balance of proteins, fats, and carbohydrates, posed a very different challenge. How could anyone replicate something as complex as milk? A product that not only nourishes, but also carries deep cultural and economic significance? The first breakthroughs came in university labs and bold startups. In 2014, companies like Perfect Day in the United States used modified microorganisms to create dairy proteins, but not full milk. The real revolution began when scientists focused on mammary cells, the same cells in cows that produce milk naturally, and succeeded in cultivating them in the lab. By 2018, the Israeli company Wilk was growing cow mammary cells to produce bioidentical milk, while BioMilk in the United States was exploring human breast milk, a development with huge implications for infant nutrition. Alongside other pioneers, these projects consolidated decades of research, and by 2020 prototypes of cultivated milk were already in existence, though the process was still expensive and difficult to scale. Overcoming these obstacles required better bioreactors, lower costs, and gaining the trust of regulators and consumers. So how exactly is this futuristic milk made? It all starts with the cells, the fundamental building blocks of life. To produce cultivated milk, scientists need mammary epithelial cells, the very same ones that secrete milk during lactation in a cow or a human. These cells are like tiny biological factories, designed by nature to produce a nutrient-rich liquid. But how do you obtain them? For cow's milk, scientists perform a biopsy on the animal's udder. This procedure is minimally invasive, similar to taking a skin sample. Under anesthesia, a small cluster of cells is removed, often no larger than a pea. Only a single sample is required, because the cells can multiply indefinitely once they are in the lab. After collection, the cells are tested to ensure they are viable and capable of producing milk. They are placed in a special medium that keeps them alive and stored in cell banks, ready to be used at any time. This step is essential because it ensures the process does not rely on repeated samples from animals, making production more sustainable and ethical from the very beginning. With the cells secured, the next step is to grow them. This happens inside a bioreactor, a machine that looks like a mix between a stainless steel tank and a high-tech aquarium. The bioreactor is the heart of the process, a carefully controlled environment where the cells can thrive. Here, scientists recreate the conditions of a living body, a constant temperature of about 37 degrees Celsius, precise oxygen levels, and a steady supply of nutrients. The cells are placed in a nutrient solution, a liquid that contains everything they need to grow: glucose for energy, amino acids to build proteins, fatty acids to form fats, and vitamins to maintain their health. This solution is like the perfect smoothie for the cells, designed to mimic the fluids that nourish them in a living organism. The bioreactors also gently stir the mixture to ensure that the cells are evenly distributed and receive oxygen uniformly. During this stage, the cells divide and multiply, forming a larger population. This process can take days or even weeks, depending on how many cells are required. Modern bioreactors are equipped with sensors that monitor pH, temperature, and other parameters in real time, ensuring that the cells remain in an optimal state. Once a sufficient number of cells has been cultivated, scientists shift the focus. They no longer want the cells to multiply, but to begin producing milk. This shift is like flipping a biological switch, and it is triggered by molecular signals similar to the hormones that induce lactation in a living body. These signals are usually a combination of growth factors and chemical compounds that tell the cells it is time to change modes. Instead of dividing, the mammary epithelial cells begin secreting milk, a liquid containing the same components as traditional milk, proteins like casein and whey, fats in the form of triglycerides, and carbohydrates such as lactose. What is fascinating is that this milk is bioidentical at the molecular level, meaning it has the same chemical and nutritional composition as milk produced by a cow. For example, cultivated cow's milk contains about 3.5% fat, 3.2% protein, and 4.8% lactose, the same as traditional milk. The secretion process happens inside the bioreactor, where the cells, either attached to a matrix or suspended in the liquid, release tiny droplets of milk into the surrounding medium. This step is the core of the technology, and recent advances have optimized it so the cells can produce milk continuously and efficiently. Companies like Wilk have refined this process, making it possible for cells to produce milk in significant quantities, though scaling production to commercial levels is still a challenge. Once the cells have secreted enough milk, the next step is collection. The liquid inside the bioreactor, which contains milk along with cell residues and leftover nutrients, is carefully extracted. This raw liquid goes through a series of filtration and purification processes to separate pure milk from any impurities. Filtration may involve high-precision membranes that remove cells, remains of the nutrient solution, and other contaminants, leaving only clean milk. At this stage, scientists can also adjust the composition of the milk according to market needs. For instance, they can remove lactose through enzymatic processes, creating a milk suitable for people with lactose intolerance. It is also possible to enrich the milk with additional nutrients such as vitamins or minerals, or adjust the fat profile to make it lighter or richer, depending on the desired final product. This level of customization is one of the great advantages of cultivated milk, as it allows the product to be tailored to specific dietary needs without compromising quality. The result is a white, creamy liquid that, when analysed, is indistinguishable from natural milk in terms of taste, texture, and nutritional value. Laboratories have reported that their cultivated milk has the same smooth, slightly sweet flavour as cow's milk, but with a much lower environmental impact. This collection and processing stage takes place under sterile conditions to ensure the milk is safe for consumption, free of bacteria or contaminants sometimes found in traditional milk. The final step is getting the milk from the lab to the consumer. Once purified, the milk is packaged in sterile conditions to prevent contamination. Depending on the final product, it can be bottled or cartoned as liquid milk, processed into powder for longer shelf life, or used as a base for other dairy products such as cheese, yogurt, or butter, since its composition supports the same fermentation and curdling processes as traditional milk. Producing milk in a laboratory may sound like science fiction, but it is already a reality that is transforming how we think about food. This process not only eliminates the need for milking animals, but also drastically reduces environmental impact. Compared to conventional dairy farming, cultivated milk uses 90% less water, 95% less land, and produces 82% fewer carbon emissions. It also provides an ethical solution by avoiding animal exploitation and makes it possible to produce milk anywhere in the world, even in regions where dairy farming is not viable. However, the road to mass adoption is not without challenges. Production costs, although they have dropped significantly, are still higher than those of conventional milk. Bioreactors and cell nutrients are expensive, and scaling production requires substantial investment. On top of that, regulators such as the FDA in the United States demand rigorous testing to guarantee product safety, and consumers still need to get used to the idea of drinking milk that was grown in a laboratory. Despite these obstacles, the market for dairy alternatives is growing quickly, with projections reaching into the billions. Now, we shift course and travel straight to Kyoto, Japan, to uncover the secrets behind matcha tea, the green powder that has conquered the world. More than 1,000 years ago, in the 8th century Japan, tea arrived from China as an exclusive luxury reserved only for monks and the nobility. But everything changed in the 12th century, when a Buddhist monk returned from China bringing not only seeds, but also a powerful philosophy. Tea as a gateway to meditation and deep spiritual connection. Zen monks embraced matcha, weaving it into their rituals, and thus was born a symbol of elegance and spirituality that we now know as the Japanese tea ceremony, the chanoyu. Back then, cultivating tea for matcha was a rudimentary but intentional process. Tea plants, Camellia sinensis, were grown on small plots in the hills of Kyoto, where the humid climate and fertile soils were ideal. Farmers already understood the importance of shading the plants before harvest, a practice that, while less sophisticated than today, made a crucial difference. They covered the bushes with straw or bamboo mats to reduce sunlight, which enhanced the flavour and deepened the colour of the leaves. This shading, though manual and labour-intensive, was key to producing tea with more chlorophyll and a distinctive umami taste. Harvesting was an art form. The leaves were hand-picked in spring, with only the youngest buds selected. They were then steamed in large wood-fired cauldrons and dried in the sun or in primitive ovens. The process culminated in grinding the leaves with hand-turned stone mills, a task that could take hours just to produce a few grams of powder. This matcha was a treasure, reserved for the elite, the temples, and the most important ceremonies. Interestingly, in feudal Japan, matcha was so valuable that it was sometimes used as a form of payment. Feudal lords accepted sacks of matcha as tribute, and possession of it was a sign of status. It was a world where a bowl of tea could be worth more than gold. Matcha was not only a product, but also a cultural expression. Samurai drank it before battles to sharpen their focus, and poets used it as inspiration. Every step, from cultivation to grinding, carried with it a nearly spiritual respect for nature. That legacy remains alive in Kyoto, where modern producers honour those traditions while adapting them to today's world. So, how is the process carried out now to produce this powerful tea? It begins in the tea fields of Kyoto, where the climate and soil remain as perfect as they were centuries ago. In mid-April, when the first tea buds emerge, farmers begin the shading process. They cover the plants with special nets that block 70 to 90% of sunlight. This step, which lasts about 3 weeks, is crucial. The shade stimulates chlorophyll production, giving matcha its vibrant emerald green colour, and encourages the creation of L-theanine, an amino acid that provides the signature umami flavour. Shading not only protects the tender buds, but also reduces photosynthesis, causing the leaves to accumulate more nutrients. A shaded leaf is darker, softer, and richer in flavour than one grown under direct sun. While synthetic nets are used today instead of straw, the method remains a faithful continuation of the monks' techniques of the past. By early May, when the buds have developed 5 or 6 new leaves, the harvest begins. This is a critical moment, since only the youngest and most tender leaves are suitable for high-grade matcha. Pickers carefully select each leaf by hand, ensuring that only the finest move onto the next stage. This work is meticulous. A skilled picker can spend hours filling a single basket, but the quality makes it worth the effort. The first spring harvest, known as ichibancha, is the most coveted. The leaves from this harvest have a fresher and more complex flavour than those from later harvests. While tea can be picked several times a year, producers use only the first harvest to guarantee an exceptional product. In some regions of Japan, traditional farmers once harvested at night under the light of the moon to prevent the sun from damaging the fresh leaves. Today, technology is used to protect them instead. Once harvested, the leaves are transported immediately to the processing plant. Time is critical, as any delay can affect freshness. The first step is to steam the leaves for 10 to 20 seconds. This process, unique to Japanese tea, halts oxidation and preserves the bright green colour and fresh flavour of matcha. The exact amount of steam is an art that requires years of experience to perfect. Next, the leaves are dried in large hot air machines. They pass through meshes that separate them and drop individually onto conveyor belts that carry them into a drying oven. Here, the leaves lose almost all their moisture, shrinking to about 20% of their original weight. The result is araka, a semi-processed tea that is stored in refrigerated chambers to preserve freshness until needed. But araka is not yet matcha. To transform it, the semi-processed leaves go through a refining process. First, they are cut to a uniform size. Then, specialized machines separate the fleshy parts of the leaves from stems and veins, a process repeated several times. An electrostatic separator removes any particles of stems or old leaves, leaving only the soft leaf tissue. This intermediate product is called tencha. Tencha is the foundation of matcha, and its quality depends on precision at this stage. Producers process only the amount needed on demand, ensuring that each batch is fresh. This way of working guarantees that matcha maintains its optimal taste and quality. The final stage is grinding, where tencha becomes matcha. Producers use traditional stone mills operated by skilled artisans. The grinding room is kept at 40% humidity with limited light to protect the leaves. Each mill produces only 30 to 40 grams of matcha per hour, a slow process that ensures a fine, micronized powder with a silky texture. Before grinding, a master blender combines different batches of tencha to achieve the perfect balance of taste, aroma, and color. The stone mills, maintained in perfect condition, extract the finest flavor from the leaves. Freshly ground matcha is immediately packaged, sealed, and sent to warehouses so it reaches customers at peak freshness. These stone mills are made of granite sourced from specific regions of Japan. Each pair can cost thousands of dollars and must be hand-carved by specialized artisans. A single mill can last decades if properly cared for. Producers are committed to delivering the freshest matcha to the cup, which is why they grind and package only on demand, shipping batches twice a month to their warehouses. This process ensures that every sip reflects the care and dedication of farmers, artisans, and tea masters. And that is how matcha tea and biotechnological cow's milk are produced. Tell me, what did you think of these processes? Let me know in the comments. If you enjoyed the video, click the like button and don't forget to subscribe to the channel. In the windows you see here, there are more videos you might enjoy, so go ahead and click one. See you next time.